April 26, 1986. Reactor 4 at the Chernobyl nuclear power plant in northern Ukraine explodes. The blast tears through the containment building, sending a plume of radioactive material 2 kilometers into the atmosphere. Fallout spreads across Europe. Contamination levels around the plant reach lethal doses within minutes. A 10 square kilometer stretch of pine forest surrounding the facility absorbs so much radiation that the trees die where they stand, their needles turning rust red before falling. They call it the Red Forest. Every animal that couldn't flee dies within weeks. Radiation poisoning, cancers, cellular breakdown. The release of radioactive material is 20 times greater than the atomic bombings of Hiroshima and Nagasaki combined. The reactor itself, still containing tons of radioactive fuel, is too dangerous to move. Engineers seal it in a concrete sarcophagus, a temporary tomb designed to contain the ongoing decay. Inside, in vast cooling pools, spent fuel rods sit submerged in water. The water itself glows an eerie blue from Cherenkov radiation, a phenomenon that occurs when particles move faster than light through a medium, splitting water molecules and releasing energy as an otherworldly luminescence. For years, the water stays clear, sterile. Nothing can survive that level of radiation, or so everyone assumes. But sometime in the early 1990s, maintenance robots sent into the reactor to inspect the fuel pools begin sending back strange images. The water isn't clear anymore. It's darkening, turning black. Something is growing in there, in water so radioactive it would kill a human in minutes. Something is not just surviving, it's thriving. 2007. Scientists finally identify the organism blackening Chernobyl's reactor pools. It's a fungus. Several species, actually. Cryptococcus neoformans, Cladosporium sphaerospermum, Fungiella dermatitidis. All of them contain high concentrations of melanin, the same pigment that darkens human skin. But these fungi aren't just tolerating the radiation. They're growing toward it, seeking it out, and most remarkably, they appear to be using it, converting gamma radiation into chemical energy through a process now called radiosynthesis. Like plants using sunlight for photosynthesis, these fungi are feeding on nuclear decay. On the most toxic byproduct of human civilization, they found a meal. The implications are staggering. If a fungus can adapt to eat radioactive waste, what else can fungi consume? Our plastic pollution choking the oceans, the carbon dioxide warming our atmosphere, the pharmaceutical contaminants in our water. We're facing an environmental crisis built from indestructible waste, and here in the ruins of Chernobyl, a fungus is demonstrating that nothing is truly indestructible. Given time, given the right organism, destruction itself can become a resource. But this raises a deeper question. Where did this ability come from? How does a simple fungus develop the capacity to metabolize uranium decay? The answer, it turns out, lies not in some recent evolutionary innovation, but in an ancient toolkit refined over billions of years. Because fungi are old, incomprehensibly old, and their story, winding back through deep time, reveals something profound about life on Earth. They're not just survivors, they're the architects, the hidden engineers who make complex life possible in the first place. And understanding where they came from might just show us how to survive what's coming next. If you appreciate this kind of deep dive into the science that shapes our world, please consider subscribing. We explore these stories carefully and thoroughly because they deserve your time and attention. Let's rewind the clock. Not decades, but hundreds of millions of years, to a time when Earth looked nothing like it does today, 410 million years ago. The Devonian period. The continents are still assembling into the supercontinent Gondwana. The climate is warm, tropical, and for the first time in Earth's history, the land is green. Plants have made the leap from ocean to shore, spreading across the barren rock in a living carpet. But these aren't the plants you'd recognize. No flowers, no grass, no trees. The tallest vegetation reaches perhaps a meter high. Primitive ferns, early mosses, spindly, delicate things clinging to thin soil. From a hilltop, the Devonian landscape looks almost like tundra. Low, sparse, vulnerable. But there's something else. Scattered across this prehistoric plain, rising above everything else, are structures that seem impossible. Towers, smooth cylindrical columns with no branches, no leaves, no visible features except their sheer overwhelming verticality. Some reach 2 meters, others 5. The largest, towering monuments to an alien biology stretch 8 meters into the Devonian sky, a meter wide at the base, tapering slightly toward the top. In a world where plants hug the ground, these things are skyscrapers, and they are not plants. 1859, Gaspé Bay, Quebec. A Canadian geologist named John William Dawson is exploring Devonian rock formations along the coast when he finds fossils of these columnar structures. Large, well-preserved, utterly baffling. Dawson is a skilled geologist, but also a committed creationist, uncomfortable with Charles Darwin's recently published theory of evolution. When faced with something strange, Dawson tends toward the familiar. He examines the fossils and concludes they're the remains of ancient conifer trees, partially rotted and preserved in unusual circumstances. He names them Prototaxites, roughly translated, first yew. But Dawson's rotted conifers present immediate problems. There's no evidence of roots, no branches, no bark texture, just smooth, featureless trunks extending from the ground like organic pillars. And they're too old. The Devonian is before true trees evolved, before plants figured out how to build wood strong enough to grow tall. These things are 8 meters high in an era when the tallest plants are barely waist height. It doesn't add up. What Dawson didn't know was that he wasn't even the first to find these fossils. 17 years earlier, in 1842, one of Darwin's close colleagues, botanist Joseph Hooker, had recovered similar specimens from Devonian rocks in Scotland. Hooker, brilliant but disorganized, mislaid the samples. They sat forgotten in a drawer until 2012, when researchers finally rediscovered and analyzed them. By then, the mystery of Prototaxites had only deepened. Because in the 160 years since Dawson's original description, paleontologists had found these structures everywhere. Canada, Scotland, Australia, Saudi Arabia. Wherever Devonian terrestrial rocks were exposed, Prototaxites appeared. They existed from 417 million years ago to 360 million years ago, 57 million years. For context, that's more than 20 times longer than modern humans have existed. These weren't rare anomalies. They were a global phenomenon, a dominant life form, and nobody could agree on what they were. The 20th century brought new theories. Maybe they were algae, some kind of marine seaweed washed ashore and rolled into logs by waves. But they're found hundreds of kilometers inland, in freshwater deposits. Maybe they were liverworts, primitive plants somehow bundled together into columns. But the internal structure doesn't match any known plant. The debate raged. Conferences were held, papers published, counterpapers written. Prototaxites became paleontology's most frustrating puzzle. Then, in 2001, a paleobotanist named Francis Huber at the Smithsonian examined thin sections of Prototaxites under high-powered microscopes and found something everyone had missed. The internal structure wasn't wood, wasn't plant cells. It was tubes, microscopic filaments woven together in a dense mat. Some tubes were narrow, barely visible. Others were wider, and they were organized in a very specific way. They branched, they fused, they intertwined at odd angles. This pattern had a name: hyphae, the structural building blocks of fungal tissue. Prototaxites wasn't a tree, wasn't a plant at all. It was a mushroom, a truly colossal mushroom, 8 meters tall, dominating the Devonian world not through leaves or photosynthesis, but through something else entirely. This revelation, published in 2007 by paleobotanist C. Kevin Boyce, resolved decades of controversy. Prototaxites was a fungus, but it also opened a far more unsettling question. How? How does a fungus grow 8 meters tall? The largest mushrooms today reach perhaps a meter. Giant puffballs in perfect conditions might hit 3 feet across. But 8 meters? That's the height of a 3-story building. In the Devonian, with thin soils and primitive ecosystems, what was feeding these monsters? And more importantly, what does their existence tell us about fungi themselves? Because if fungi could dominate an entire planet for 57 million years, growing taller than anything else alive, then they're not the minor players we've assumed them to be. They're something far more powerful and far more ancient. To understand what made Prototaxites possible, we need to look at what fungi actually are. Not the mushrooms we see, which are just fruiting bodies, sexual organs meant to spread spores, but the real organism, the body hidden underground. And for that, we travel to Oregon. The Malheur National Forest in eastern Oregon is a landscape of rolling hills covered in ponderosa pine, popular with hikers, hunters, families camping under clear alpine skies, peaceful, unremarkable. Except that beneath the forest floor, spread across nearly 10 square kilometers, lives the largest organism on Earth. Not the largest animal, not the largest plant, the largest individual, period. And most hikers walk right over it without knowing it's there. 1998. A team of forest pathologists is studying a problem. Trees in Malheur are dying, not from beetles or drought, but from a fungal infection. In autumn, dark, honey-colored mushrooms sprout around the bases of infected pines, beautiful in an ominous way. But by the time the mushrooms appear, the tree is already dead. The fungus has been eating it from the inside, infiltrating the roots, working its way up through the sapwood, blocking water transport and nutrient flow. The mushrooms are just the final announcement. This tree belongs to us now. The fungus is Armillaria ostoyi, honey mushroom, a well-known pathogen that foresters dread because once it establishes in a forest, it's almost impossible to eradicate. But what the Oregon researchers wanted to know was how far the infection had spread. So they started taking samples. From infected tree to infected tree, they collected pieces of the fungal tissue, those white thread-like filaments growing through the roots and soil. Back in the lab, they conducted 2 tests. First, they placed tissue samples from different trees together in petri dishes. If the samples were from separate fungal individuals, they would remain distinct. If they were from the same organism, they would fuse together, recognizing each other as self. Sample after sample fused. Every tree they tested across kilometers of forest contained genetically compatible tissue. To confirm, they extracted DNA, and the results were unmistakable. Every sample was genetically identical, not similar, identical. Like cells from your left hand and right hand sharing the same genome because they're part of the same body. Across 2,384 acres, the Armillaria fungus was one individual, one organism. Older than the trees it was killing, older than the forest itself. Based on growth rates, researchers estimated its age at 2,400 years old, possibly twice that. It had been alive since the Roman Empire, through the rise and fall of civilizations, through wars, plagues, the Industrial Revolution, 2 world wars, the moon landing. This single fungus, spreading silently beneath Oregon soil, had witnessed 100 human generations. And here's what makes it truly alien. That massive organism, covering thousands of acres, isn't a solid mass. It's a network, a web of microscopic threads called mycelium, each thread thinner than a human hair, weaving through soil and wood and leaf litter in a 3-dimensional mesh so dense that a single teaspoon of forest dirt can contain up to 10 kilometers of these filaments. 10 kilometers in a teaspoon. The mycelial network is like a biological internet, connecting distant parts of the forest, transmitting information, sharing resources, responding to threats. Except this internet is alive, and it's been running for thousands of years. But the Oregon fungus, massive as it is, isn't unique. It's just the largest one we've found. Mycelia like this exist in forests worldwide, under every woodland, every meadow, every patch of soil rich enough to support complex life. They're invisible, easy to dismiss. But remove them, and the entire ecosystem collapses. Because mycelium doesn't just feed itself, it feeds everything else. And to understand how, we need to look at what these filaments are actually doing down there in the dark. Mycelium is made of hyphae, individual fungal threads, each about 5 microns across, 1/20th the width of a human hair. A single hypha is 1 cell thick, just a tube. A cellular pipeline filled with cytoplasm and organelles, surrounded by a wall made of chitin, the same material in insect exoskeletons. But this tube has a trick. At its tip, the cell wall is softer, more flexible, and the hypha pushes forward, exploring soil the way a plant root explores, except faster. A growing hyphal tip can extend several millimeters per hour, branching every few millimeters, creating a fractal network of exploring fingers. When millions of hyphal tips grow simultaneously, the mycelial network expands like a living search engine, probing every cubic centimeter of soil, looking for food. And when it finds food, it doesn't swallow. It digests externally. The hyphae secrete enzymes, powerful chemical cocktails that break down organic matter outside the fungal body. Dead leaves, fallen branches, animal carcasses, even rock. The enzymes shatter complex molecules into simpler nutrients, which the hyphae then absorb. This external digestion allows fungi to eat things no animal could stomach. Wood, lignin, cellulose, chitin, keratin, materials that would pass through an animal's gut unchanged. Fungi dismantle them at the molecular level. They're the ultimate recyclers, nature's garbage disposal. Without them, dead organic matter would pile up indefinitely. Forests would choke on their own shed leaves. But fungi break it all down, releasing nutrients back into the soil, keeping the ecosystem moving. But here's where it gets strange. That mycelial network, those billions of hyphal tips exploring soil, don't operate independently. They communicate, coordinate, make collective decisions. In 2000, a Japanese researcher named Toshiyuki Nakagaki conducted an experiment that changed how scientists think about fungal intelligence. He took a species of slime mold, technically not a fungus, but close enough in lifestyle, and placed it in a maze. At one end, he put a piece of oat flake, food. The slime mold, starting from the other end, began exploring the maze, sending out tendrils through every corridor. Dead ends were explored and abandoned. Successful paths were reinforced. Within hours, the organism had found the shortest route from start to food, abandoning all inefficient branches. Nakagaki repeated the experiment with a map of Tokyo and its subway system, placing oat flakes at major station locations. The slime mold recreated the Tokyo subway network, not perfectly, but efficiently, connecting all stations with minimal redundancy. It solved an engineering problem that had taken humans decades of planning. Fungi aren't conscious. They don't have brains, don't think in any way we'd recognize. But the mycelial network behaves like a distributed intelligence. Information from millions of hyphal tips is integrated. Decisions are made. Grow here, retreat there, reinforce this connection. The network rewrites itself constantly, optimizing for efficiency. And the communication between distant parts of the mycelium appears to be electrical, not chemical signals, which are slow, but electrical pulses traveling through the hyphae at speeds approaching nerve transmission in animals. Some mycologists have started calling mycelium a fungal brain, not metaphorically, structurally, a network that processes information, stores patterns, and responds adaptively to environmental change. Whether that constitutes intelligence is a philosophical question. But functionally, it works, and mycelium is strong. Absurdly strong for something made of threads thinner than hair. There are documented cases of mushrooms breaking through asphalt roads as they grow, cracking pavement that weighs tons. In laboratory experiments, oyster mushrooms have been observed growing through Kevlar, the material used in bulletproof vests. The pressure generated at a hyphal tip can reach 8 atmospheres, 8 times sea level air pressure, focused on a 0.5 microns wide. That's enough to penetrate plant roots, to work between grains of rock, to literally reshape the substrate it grows through. Over time, fungal hyphae shatter stone, creating the first soils on barren landscapes. They're not just biological organisms, they're geological forces. But the most profound thing mycelium does isn't digesting dead matter or cracking rocks. It's partnership. Because fungi don't just take, they give. And in doing so, they've fundamentally shaped the evolution of life on land. 1997, University of British Columbia. A forest ecologist named Suzanne Simard is studying Douglas fir and paper birch trees in the interior rainforests of British Columbia. She's interested in competition. Foresters have long assumed that different tree species compete for resources: sunlight, water, nutrients. Standard Darwinian competition. Simard wants to test this. So she designs an experiment. She injects paper birch trees with carbon-14, a radioactive isotope. Then she waits. If trees compete, the carbon-14 should stay in the birch, hoarded against competitors. But that's not what happens. Within days, the radioactive carbon appears in neighboring Douglas fir trees. Not through the air, not through leaf contact, through the soil, through the mycorrhizal network connecting their roots. The fungi colonizing birch roots are the same species colonizing fir roots, creating a living bridge between trees. And carbon is flowing across that bridge, from birch to fir. CIMAD injects fir trees. Carbon flows to birch. The trees are sharing, feeding each other through the fungal network. But it's more than just carbon. CIMAD continues her research for decades, and the picture that emerges is extraordinary. The mycorrhizal network, that web of fungal hyphae connecting tree roots across entire forests, isn't just a passive transport system, it's an active communication network. When one tree is attacked by insects, it releases chemical signals into the mycelium. The fungi carry those signals to neighboring trees, which respond by producing defensive compounds before the insects arrive. The forest is sharing information, warnings, preparation for threats not yet present. And the sharing isn't random. Older trees, what CIMAD calls mother trees, act as hubs in the network. A single large tree can be connected to dozens of smaller trees, funneling carbon and nutrients to shaded saplings struggling to photosynthesize. The forest isn't a collection of individuals competing for resources. It's a cooperative, an interdependent network where success isn't measured by individual dominance, but by collective survival. And the fungi make it possible. This discovery, published throughout the late 1990s and 2000s, fundamentally changed forest ecology. What CIMAD called the Wood Wide Web became a new paradigm. Forests aren't just trees. They're integrated systems, superorganisms, and fungi are the neural network holding them together. Without mycorrhizae, trees couldn't access enough nutrients from the soil. Their roots, thick and limited, can't penetrate every crevice. But fungal hyphae can. They slip between soil particles, extracting phosphorus, nitrogen, micronutrients that roots can't reach. In exchange, trees provide the fungi with sugars from photosynthesis. It's a trade, carbon for nutrients, and it's not optional. 90% of land plant species form mycorrhizal partnerships. Remove the fungi, and most plants can't survive. But here's the truly remarkable part. This partnership isn't new. It's not something that evolved recently as forests became complex. It's ancient, older than trees, older than flowers, older than almost everything we think of as terrestrial life. Because when plants first crawled out of the ocean 450 million years ago, they didn't have roots. And they couldn't have survived without help. The first land plants were tiny, fragile, millimeters tall. They had no vascular tissue to transport water, no root systems to anchor them or gather nutrients. The soil, such as it was just weathered rock and bacterial mats. No organic matter, no nitrogen cycle, no established ecosystems. It should have been impossible for plants to survive. And yet they did, because they had partners. In 2010, paleobotanist Christine Struludarian examined fossilized plant roots from 450 million years ago and found something inside them. Mycorrhizal fungi, the same structures CIMAD was studying in modern forests. Except these were in the earliest land plants, which means fungi were there first, or at least they arrived together. And for the first 50 million years of terrestrial plant life, Fungi acted as surrogate roots. Plants photosynthesized. Fungi gathered nutrients. Together, they transformed a barren planet into a green one. This is what made Prototaxites possible. Not competition, cooperation. The Devonian world was a place where fungi and plants were still figuring out how to coexist. And in that transitional period, fungi, with their vast mycelia and external digestion, had a temporary advantage. They could grow tall, not because they were stronger, but because they didn't need to photosynthesize. They fed on the dead, on decay, on the recycling of nutrients in an ecosystem still learning how to function. And for 57 million years, they were kings. But eventually, plants caught up, evolved true roots, vascular tissue, wood, and trees took over the vertical space. Prototaxites vanished from the fossil record, outcompeted, or perhaps just no longer necessary. The giant mushrooms collapsed back into the soil, where fungi have remained ever since. Hidden, unseen, but no less essential. Which brings us back to the question. If fungi have been partnering with plants for 450 million years, enabling their success, feeding them, connecting them, then how far back do fungi themselves go? When did they first appear? What was the first fungus on Earth? The answer is older than you think. And stranger. Ты прав, первая часть получилась короткой. Продолжаю вторую часть про грибы, углубляясь в их эволюцию, связь с человечеством, древние окаменелости и будущее. Пишу дальше. But to answer that question, we need to step away from the forests and the fossil record for a moment. Because fungi's relationship with complex life isn't limited to plants. It extends to us, to humans. And that connection is far older, far stranger, and far more intimate than most people realize. 1487, Tenochtitlan, the capital of the Mexica Aztec Empire, one of the largest cities the world has ever seen. Built on an island in the middle of Lake Texcoco, connected to the mainland by causeways, the city is a marvel of engineering and urban planning. Aqueducts bring fresh water from distant springs. Chinampas, floating gardens, produce food for hundreds of thousands of people. At the city's heart rises the Templo Maya, a massive pyramid temple dedicated to the gods of war and rain. And on this particular occasion, the temple is the site of something extraordinary and terrible. The new emperor, Ahuitzotl, one of the most formidable military leaders in Mesoamerican history, is consolidating power. He's just completed a series of successful military campaigns, expanding the empire into new territories. And to celebrate, to demonstrate the might of the Mexica and the favor of the gods, he orders a ceremony, a rededication of the Templo Maya. The festivities last 4 days. And during those 4 days, according to Spanish friar Diego Duran, writing 2 generations later, tens of thousands of captive warriors are brought to the top of the pyramid. One by one, their hearts are cut from their chests while still beating, offered to the gods in an orgy of ritual sacrifice. Duran, writing in the 1580s, was one of the few Spanish chroniclers who actually spoke with surviving Aztec elders, recording their histories before they were lost to colonial suppression. And he describes something else that happened during those 4 days. For those not participating in the sacrifices, the extended coronation festival was cause for celebration. Chocolate was drunk, songs were sung, dances performed, and a very special delicacy was consumed. The Aztecs called it Teonanacatl, flesh of the gods, a small spindly mushroom with a white stem and golden cap. Celebrants ate it raw, sometimes with a little honey. and within minutes they were transformed. Some found themselves laughing uncontrollably, dancing without reason. Others experienced disturbing visions, wars, demons. Their own deaths played out before their eyes. Some wept openly, overcome with emotion. Others retreated quietly to their homes, meditative and introspective. And afterward, they gathered to discuss what they had seen. They believed the mushrooms were a gift from Quetzalcoatl, the feathered serpent god, that consuming them allowed communion with the divine, that the visions were messages, prophecies, spiritual truths revealed through the flesh of the gods. The Spanish conquistadors who arrived decades later were horrified by these practices, not just the human sacrifice, which they condemned as barbarism, but the use of mind-altering mushrooms in religious ceremonies. They saw it as devil worship, witchcraft. The colonial authorities made great efforts to erase all records of these substances and the cultures that used them. After the fall of the Aztec Empire, knowledge of the sacred mushrooms faded. Their very existence became controversial. Had they even been real? Or were they myths, invented by traumatized chroniclers trying to make sense of a civilization so alien to European experience? Samples sent to Harvard in the early 20th century arrived too rotten to study. For decades, Western scientists debated whether psychedelic mushrooms were real or folklore. It wasn't until the 1950s that adventurers retracing Duran's steps into the mountains of Oaxaca witnessed a modern Mazatec mushroom ceremony led by a curandera named Maria Sabina. The mushrooms were real, and they were exactly as powerful as the Aztecs had claimed. In 1958, Swiss chemist Albert Hofmann, the same man who had synthesized LSD 15 years earlier, isolated the active compound from the sacred mushrooms. He called it psilocybin, a tryptamine molecule that mimics serotonin in the human brain, binding to receptors and producing profound alterations in perception, emotion, and cognition. Under its influence, users report synesthesia, seeing sounds and hearing colors, ego dissolution, a sense that the boundary between self and world has dissolved, mystical experiences indistinguishable from religious revelation, time distortion, geometric hallucinations, the feeling of profound interconnectedness with all living things. And the Aztecs were far from the first to discover this. Ancient manuscripts predating the Spanish conquest depict feathered deities holding mushrooms. Stone sculptures shaped like mushrooms, dated to the 2nd millennium BCE, have been found across Mesoamerica. In Algeria, cave paintings created between 7000 and 9000 BCE show a horned figure, possibly a shaman, with mushroom shapes sprouting from its shoulders and arms. In classical Greece, the Eleusinian mysteries, secretive ceremonies attended by Plato, Aristotle, and countless other influential thinkers, are theorized to have involved ergot, another psychedelic fungus that grows on rye. For as long as humans have been human, we've been eating mushrooms that change our minds. In 1992, ethnobotanist Terence McKenna proposed what he called the stoned ape theory. It's a fringe idea, not accepted by mainstream anthropology, but it's provocative enough to consider. McKenna observed that human culture, language, art, and social complexity seemed to explode suddenly in the archaeological record around 50,000 to 70,000 years ago. Before that, hominids used simple tools. After, we see cave paintings, jewelry, elaborate burials, long-distance trade networks. Something changed. McKenna suggested that what changed was diet, that early humans foraging in African grasslands encountered psilocybin mushrooms growing in dung, that consuming them regularly produced cognitive changes, enhanced pattern recognition, increased visual acuity, greater introspection and social bonding. And over generations, these effects shaped human evolution, expanding our minds and making modern consciousness possible. There's almost no way to prove this. The fossil record doesn't preserve mushroom consumption. But we do know that fungi have been manipulating animal behavior for millions of years. Cordyceps fungi infect insects, taking over their nervous systems and forcing them to climb to high locations before the fungus erupts from their bodies to spread spores. Fossilized leaves from 48 million years ago show evidence of cordyceps infection, meaning fungal mind control is at least that old. If fungi can rewire an ant's brain to serve fungal reproduction, is it so implausible that psilocybin mushrooms could have influenced human cognition? Not through deliberate manipulation, but through the simple fact that humans who ate them gained advantages. Better pattern recognition means better hunting. Stronger social bonding means more cohesive tribes. And tribes that thrive pass on their behaviors, including the ritual consumption of mind-altering fungi. Whether or not McKenna's theory holds, one thing is undeniable. Humans and fungi have co-evolved. Some yeasts live only on the human skin, adapted so completely to our bodies that they can't survive elsewhere. When researchers analyzed Neanderthal dental plaque, they found traces of Mycelium mold ground into the teeth, likely used to treat infections. The same practice was known among indigenous Australians and ancient Egyptians. Otzi, the Iceman, the 5,000-year-old mummy found frozen in the Alps, carried pieces of polypore fungus in his kit, probably for starting fires and treating wounds. Medieval chroniclers used ink made from ink-cap mushrooms to write on parchment. And of course, uncountable tons of edible mushrooms have sustained human populations throughout history, despite being feared and avoided in many cultures. In 1928, Alexander Fleming accidentally rediscovered penicillin, the antibiotic produced by Mycelium mold. He left a petri dish uncovered, and mold contaminated it. When he returned, he noticed that bacteria near the mold had died. That accidental observation became one of the most important medical discoveries in history. By World War II, penicillin was being mass-produced, saving millions of lives. Infections that had been death sentences became treatable. Battlefield wounds that would have festered and killed soldiers instead healed. The Allies' ability to produce penicillin in large quantities arguably helped turn the tide of the war. And that life-saving drug came from a fungus. Today, 70% of antibiotics are derived from fungi. We owe at least part of our success as a species to these organisms. But fungi do more for us than medicine and food. They do something far more fundamental, something we're only beginning to understand. Remember Suzanne Simard's Wood Wide Web, that mycorrhizal network connecting trees in a forest? It's not just trees. The same networks connect grasslands, meadows, agricultural fields. Anywhere plants grow, fungi are underneath, weaving them together. And these networks don't just transport carbon and nutrients, they transmit warnings. In experiments conducted in the 2000s, researchers found that when one plant is attacked by aphids, it releases chemical signals into the mycorrhizal network. Neighboring plants, connected by the same fungi, receive those signals and begin producing defensive compounds before the aphids arrive. The plants are communicating through fungal intermediaries. But it goes further. In some forests, carbon flows not just between trees of the same species, but between different species, birch to fir, oak to pine, even between trees and understory shrubs. And the direction of flow isn't random. Carbon moves from areas of abundance to areas of scarcity, from sunlit canopy trees to shaded saplings struggling to photosynthesize. This isn't competition, it's altruism, enforced or at least facilitated by the fungal network. Why would a tree give resources to a competitor? It doesn't, directly. But the fungi connect them, and perhaps the fungi, feeding on both, benefit from keeping the entire network healthy. A forest with diverse, healthy trees produces more organic matter for fungi to decompose. So the fungi act as mediators, redistributing resources to maximize total ecosystem productivity. It's not cooperation in the sense of conscious choice, but it functions as cooperation. And it creates ecosystems far more resilient than any collection of individuals competing for scarce resources could be. This realization, which has emerged over the last 2 decades, fundamentally changes how we think about ecology. Ecosystems aren't battlegrounds where individuals fight for survival. They're networks, collectives. And fungi are the architects of those networks. They make cooperation possible, not through altruism, but through symbiosis. By connecting organisms that would otherwise be isolated, fungi create interdependence. And interdependence, over evolutionary time, becomes partnership. The most extreme example of this is lichens. If you've ever seen greyish-green crusty growths on rocks, tree bark, or stone monuments, you've seen lichens. They look like plants, or maybe colonies of algae. But they're neither. Lichens are composite organisms, a partnership between fungi and photosynthetic algae or cyanobacteria, so intimate that they function as a single entity. The algae photosynthesize, producing sugars. The fungi provide structure, protection, and water retention. Together, they can survive in places where neither could live alone. Arctic tundra, desert rocks, Antarctic stone, the sides of buildings in polluted cities. Lichens even survived exposure to space. In experiments aboard the International Space Station, lichen samples were bolted to the outside of the station, exposed to vacuum, cosmic radiation, and temperature extremes. After 18 months, they were brought back inside, and they resumed growing, as if nothing had happened. Lichens aren't rare. They cover roughly 8% of Earth's terrestrial surface, more area than tropical rainforests, and they've been around for a very long time. But here's the strange part. A lichen isn't one organism. It's usually 3 or more. When researchers started using genetic techniques to study lichen DNA, they expected to find 2 genomes, fungus and alga. Instead, they found multiple fungal species, sometimes yeast, sometimes bacteria. Every lichen is a cooperative of organisms, each contributing something the others can't provide alone. And the boundary between individual and collective becomes meaningless. In 1869, Swiss botanist Simon Schwendener proposed the dual hypothesis of lichens. That they were fungi enslaving algae, forcing them to produce sugars in exchange for shelter. It was one of the first descriptions of symbiosis in nature, and it was controversial. Many scientists rejected it. Enslavement seemed too sinister, too unplant-like. But Schwendener was right, more or less. Except the relationship isn't slavery, it's partnership. The algae benefit, the fungi benefit, and together they dominate niches neither could exploit alone. Today, we know that many lichens are even more complex. Some incorporate nitrogen-fixing cyanobacteria, providing fertilizer for the partnership. Some include yeast that produces toxins to defend against herbivores. The deeper scientists probe, the more partners they find. And no one has yet determined where the lichen ends and the environment begins. This blurring of boundaries, this collaborative existence, isn't an exception in the fungal kingdom. It's the rule. Fungi don't exist in isolation. They integrate with other organisms, shape their evolution, enable their success, and in doing so, they've fundamentally altered the trajectory of life on Earth, not through competition, through partnership. Which brings us back to the question we started with. If fungi have been enabling complex life for hundreds of millions of years, partnering with plants, animals, and even humans, then understanding their origins becomes crucial. Because those origins might tell us something profound about how life works, about cooperation and symbiosis as evolutionary drivers, about intelligence without brains, about resilience through flexibility. So let's go hunting. Let's go back in time, deeper than the Devonian, deeper than the first forests, and ask, where do fungi come from? The challenge is that fungi don't fossilize well. Their bodies are soft. Hyphae are microscopic. Mushrooms decay within days. The vast majority of fungal life leaves no trace. So paleontologists have to get creative. They look for chemical signatures, for structures that can only be explained by fungal activity, for tiny filaments preserved in extraordinary circumstances, and occasionally they get lucky. Rhynie, Scotland, 1912. Workers quarrying stone in Aberdeenshire uncover a layer of chert, a type of rock formed when silica-rich water floods an area and rapidly mineralizes everything it touches. The Rhynie chert, as it becomes known, is special. It's Devonian in age, about 410 million years old. And it's a snapshot, a moment frozen in exquisite detail. When researchers examine thin sections of Rhynie chert under microscopes, they see cells, individual plant cells, spores, and hyphae. Fungal filaments preserved in 3 dimensions, their walls intact, their branching patterns visible. For paleontologists, it's like finding a photograph from the Devonian, not a blurry impression, a high-resolution image. And what the Rhynie chert reveals is staggering fungal diversity. There are arbuscular mycorrhizae, the same kind that form partnerships with plant roots today. There are saprotrophs, fungi feeding on dead organic matter. There are parasites, attacking living plants. All the major fungal lifestyles are already present, already specialized, which means fungi didn't just arrive in the Devonian. They'd been evolving for a long time before that, diversifying, adapting, becoming the ecological architects they remain today. But 410 million years is still relatively recent in Earth's history. The planet is 4.54 billion years old. Life has existed for at least 3.5 billion years. So where in that immense span of time did fungi emerge? To answer that, we need to leave Scotland and travel to Central Africa, to the Democratic Republic of Congo. Just outside the city of Mbuji-Mayi, in one of the most troubled regions on Earth, geologists have been drilling. The area is rich in diamonds, discovered in 1909, and mining has torn open the landscape. Red soil, scarred hillsides, rivers running with rusty sediment. But beneath the industrial devastation, there are ancient rocks, much older than the diamonds. And in 2010, an international team of scientists led by Urania Laurent decided to examine them. The drill core, designated Kansuki B13, penetrates limestone and shale laid down in a warm, perennial lake during the Neoproterozoic era, between 810 and 715 million years ago, nearly twice as old as the Rhynie chert. In these rocks, paleontologists found the usual suspects: spherical microfossils, probably primitive eukaryotic algae, single cells with nuclei, unremarkable for rocks of this age. But at a depth of over 100 meters, they found something else: filaments, tiny threads less than 10 microns across, 1/10 of a millimeter, but up to a millimeter in length. And they weren't randomly scattered. They were organized, branching, fusing, forming interconnected networks. In shape alone, they resembled hyphae. But at nearly twice the age of the oldest confirmed fungi, morphology wasn't enough. So the researchers employed a technique called Raman spectroscopy. You shine a laser at a sample, and the way the light scatters reveals the molecular structure. The filaments contained organic material, carbon compounds that had undergone the same heating and compression as the surrounding rock, which meant they were as old as the limestone. Not modern contamination, not secondary infiltration, original. Next, they used a chemical stain, the same stain used by soil mycologists to identify fungi in modern ecosystems. It binds to chitin, the polymer that makes up fungal cell walls, and nothing else. Under the right kind of microscope light, chitin glows green. The researchers treated the Neoproterozoic filaments with the stain, and they glowed green, which strongly suggested that chitin was part of their original structure. Combined with the morphology, the chemical composition, the age, the evidence pointed in one direction. These were fungi, Mycelia, living in the sediment of a lake 715 million years ago, feeding on decaying organic matter from algae and bacteria, a lifestyle almost identical to fungi today. 715 million years ago, Earth was a very different place. The supercontinent Rodinia was breaking apart. Ice ages were beginning, the first of the snowball Earth glaciations that would nearly freeze the planet solid. Oxygen levels in the atmosphere were rising, but still far below modern levels. And complex life was just beginning to experiment with multicellularity. The first animals, simple sponge-like creatures, were appearing in the oceans. Plants wouldn't colonize land for another 250 million years. But fungi were already there, in the lake's sediments, quiet, unnoticed, doing what they still do, breaking down the dead, recycling nutrients, making ecosystems possible. But even 715 million years might not be the beginning. In the far north of Canada, in the Northwest Territories, on the desolate frozen shores of the Arctic Ocean, geologists have found shales dated to around 1 billion years old, 200 to 300 million years older than the Congo samples. And in these shales, using traditional micropaleontological methods, dissolving the rock away and examining the organic material left behind, researchers have found structures, large spheres, 3 to 8/100ths of a millimeter across, attached to narrow filaments that branch at right angles. The interpretation? Fungal spores and hyphae. The spheres are reproductive structures. The filaments are the mycelial network. Chemical analysis suggests chitin in the walls, though the data is less conclusive than the Congo samples. If this interpretation is correct, then fungi existed 1 billion years ago, during the late Mesoproterozoic, a time when Earth's surface was still largely barren, when the most complex life was single-celled eukaryotes and bacterial mats, when the atmosphere was still adjusting to oxygen. Fungi were already present, already branching, already forming networks. 1 billion years is an almost incomprehensible span of time. It's 1/4 of Earth's total history. It's before plants, before animals, before anything we'd recognize as familiar life. And yet fungi were there, doing fungal things. Which raises a question. How much older could they be? In 2017, paleontologists from Sweden and Australia published a paper describing structures found inside volcanic rock from South Africa, basalt specifically. Ancient lava flows that erupted 2.4 billion years ago. Inside bubbles in the basalt, vesicles formed when gases tried to escape through the cooling magma. The researchers found filaments, the right size for hyphae, branching and fusing in mycelial patterns. They interpreted them as fungi 2.4 billion years ago. That's not just old, that's ancient beyond comprehension. It's early Proterozoic, barely 500 million years after the Great Oxidation event, when cyanobacteria first started producing oxygen and fundamentally transforming Earth's atmosphere and oceans. Complex eukaryotic cells were just beginning to evolve. And according to this paper, fungi already existed. The claim is controversial. Most mycologists are skeptical. The structures are inside volcanic rock, which makes chemical analysis nearly impossible. The heat and pressure have destroyed any organic material that might have confirmed fungal identity. All that remains is morphology. And morphology alone, especially at this age, isn't enough to make a definitive claim. The structures could be fungi, or they could be bacterial mats, or mineral artifacts, or something else entirely. Without supporting chemical evidence, the scientific community remains unconvinced. But even if the South African structures aren't fungi, they show something important. The mycelial growth pattern, filaments branching and fusing into networks, is very old. Older than fungi, perhaps. Maybe bacteria or early eukaryotes experimented with this lifestyle long before the fungal kingdom made it their own. Maybe the mycelial strategy is so effective, so advantageous for extracting nutrients from substrates, that it evolved multiple times independently. Convergent evolution. Different lineages arriving at the same solution. What we can say with confidence is this. Fungi, as we define them today, organisms with chitin-walled hyphae forming mycelia, probably emerged sometime between 1 billion and 750 million years ago. During the Neoproterozoic, a time of tremendous change, Rodinia breaking apart, snowball Earth glaciations, oxygen rising, and the first animals evolving. Fungi were part of that story, maybe even a driving force. Because as plants would later discover, having fungal partners makes survival on land possible. And perhaps early animals, simple filter feeders and grazers, benefited from fungal activity too. Fungi breaking down organic matter, releasing nutrients, creating soils, making the substrates where other life could thrive. For much of the last billion years, fungi have been one step ahead. Everywhere life tries to go, fungi arrive first. Or at least they're close behind, preparing the way. And that pattern continues today. In the most unlikely, most hostile environments on Earth, fungi are there. Which brings us back to Chernobyl. back to the black fungus growing in the reactor, feeding on radiation. And it forces us to ask, what else can fungi do? What other impossible environments can they colonize? What other waste can they consume? Because we're facing a crisis. Plastic pollution. 8 million tons enter the oceans every year. It doesn't biodegrade, doesn't break down. It fragments into microplastics, which infiltrate every ecosystem, every food chain, including ours. We're drowning in indestructible waste, and we don't know how to stop it. But in 2011, researchers exploring the Amazon rainforest discovered something. A fungus, Pestilotiopsis microspora, that eats polyurethane, one of the most common plastics. Normally, polyurethane takes 1,000 years to degrade. This fungus breaks it down in weeks. It secretes enzymes that shatter the polymer chains, turning plastic into food. In 2017, another species, Aspergillus tubangensis, was found in Pakistan. It eats polyester. And there are others, fungi that can metabolize PVC, polystyrene, PET, the same plastics choking our oceans. The potential is staggering. If we can cultivate these fungi, scale up their activity, we could bioremediate plastic waste. Not by inventing new technology, but by harnessing biology that already exists. Fungi have been breaking down complex organic molecules for 1 billion years. They're the ultimate recyclers. And now, faced with synthetic polymers, they're adapting, finding ways to eat what we thought was inedible. In 2020, a company called Ecovative began producing packaging materials made from mycelium. You grow fungi in molds, let the hyphae form dense networks, then heat treat them to stop growth. The result is a material that's lightweight, strong, insulating, and completely biodegradable. It can replace styrofoam, plastic packaging, even building insulation. When you're done with it, you can compost it, bury it. The mycelium breaks down in weeks. No waste, no pollution, just a return to soil. This is what fungi do. They turn endings into beginnings, death into life, waste into resources, and they've been doing it for 1 billion years. But there's one more frontier, one more impossible environment where fungi might make the difference. Space. 2016. The International Space Station. Astronauts conduct an experiment. They place samples of fungi outside the station, exposed to vacuum, cosmic radiation, temperature extremes. Conditions that would kill almost any Earth organism instantly. And they leave them there. For months. When they bring the samples back inside, they expect to find dead tissue, desiccated, destroyed. Instead, the fungi are alive, not just surviving, growing, thriving, growing 3 times faster than controlled samples inside the station. The radiation, which should have been lethal, was feeding them, just like Chernobyl. This discovery has profound implications. If fungi can survive and grow in space, they can help us live there. NASA is funding research into mycelium-based construction. The idea is simple. You send fungal spores and growth medium to Mars. Add water. The fungi grow, forming dense mycelial mats. You shape them into bricks, panels, structures. Then heat treat to stop growth. The result is a building material that's self-assembling, lightweight, radiation shielding, and biodegradable when you're done with it. You don't need to launch heavy materials from Earth. You grow them on Mars. And it goes further. Mars has no soil, just regolith, powdered rock. Plants can't grow in it, but fungi can. Fungi break down rock, secrete acids that dissolve minerals, create the first soils, just like they did on Earth 450 million years ago. If we want to terraform Mars, If we want to turn it into a living world, we'll need fungi to prepare the ground, to make it habitable, to do what they've always done, turn barren rock into ecosystems. This is the full circle. Fungi pioneered land on Earth, enabled plants, enabled animals, enabled us. And now, as we prepare to become a multiplanetary species, we're realizing we can't do it without them. We need their partnership, their resilience, their ability to turn death into life. We need fungi. And maybe that's the real answer to the question we started with. What was the first fungus? We don't know exactly. Somewhere between 2.4 billion and 700 million years ago, mycelial life emerged. Branching filaments, chitin walls, external digestion, the fungal toolkit. And once it appeared, it never stopped. It adapted, diversified, partnered with every other kingdom of life. And through that partnership, it shaped the planet, turned a barren rock into a living world. Fungi don't conquer. They compose. They take what's broken, what's dead, what's discarded, and they remake it. They're the ultimate optimists. Every catastrophe is an opportunity. Every mass extinction creates new niches. Every human mistake, from nuclear meltdowns to plastic pollution, becomes a resource. Given time, given the right fungus, and time is what they have. 1 billion years of evolutionary experimentation, 1 billion years of adapting to every environment Earth can produce. Ice ages, deserts, ocean floors, volcanic vents, radioactive waste. They've survived it all, and they'll survive what's coming. Climate change, ecosystem collapse, the Anthropocene extinction. Fungi will outlast us. They always do. And when we're gone, when our cities crumble and our plastics fragment and our monuments erode, fungi will be there, breaking it down, turning our endings into new beginnings, just like they have for 1 billion years. That's not pessimism, it's realism, but it's also hope. Because if fungi can recycle nuclear waste and plastic and dead planets, maybe they can help us too. Maybe the solution to the crises we've created isn't more technology, more industry, more extraction. Maybe it's partnership, learning to work with the organisms that have been solving these problems since before animals existed. Letting fungi teach us how to turn waste into resources, how to build without destroying, how to live in balance with a planet that, for all our arrogance, we still barely understand. The first fungus appeared over 500 million years ago. Maybe much longer, and it's still here, beneath our feet, inside our bodies, in the soil and the air and the water, invisible, overlooked, underestimated, but essential. More essential than we ever imagined. So the question isn't what was the first fungus. The question is, what will fungi make possible next? Because they're not done. They're never done. And as long as there's death to recycle, waste to consume, barren ground to colonize, fungi will be there. One step ahead, preparing the way, composing new worlds from the ruins of old ones. And maybe, if we're smart enough to listen, they'll show us how to do the same.